Imagine a root that can kill you with cyanide if you eat it wrong. Yet it feeds more than 800 million people today and has sustained communities for millennia. This is cassava, also called manioc or yucca. Raw or poorly prepared, its flesh releases hydrogen cyanide that blocks cellular respiration. Properly processed, it becomes a caloric staple that thrives in poor soils and drought. How did humans ever decide this plant was food? The answer is not luck. It is generations of observation, trial, and knowledge passed down under the constant threat of poisoning. If this kind of verified food history keeps you watching, hit subscribe. Because what follows is the documented science and survival story behind one of the most paradoxical crops on Earth. Cassava, Manihot esculenta, was domesticated from wild populations of Manihot esculenta subspecies Flabellifolia along the southern border of the Amazon basin, in what is now southwestern Brazil and eastern Bolivia. Genetic studies using single-copy nuclear genes, published in the Proceedings of the National Academy of Sciences, place the origin of domestication in this region. Archaeological evidence includes manioc phytoliths on stone tools at the Teotonio site in Brazil, dated to approximately 6,000 calendar years before present, found alongside other early domesticates in dark earth soils. Broader starch and pollen records support cultivation beginning as early as 8,000 to 10,000 years ago. Indigenous peoples of the Amazon and surrounding savannas selected varieties that produced larger, starch-rich roots. The plant solved a critical problem: reliable calories in environments where maize or other grains were less dependable. It spread across the neotropics long before European contact. Portuguese traders carried it from Brazil to Africa in the 16th century, where it was adopted first as a famine reserve, and later as a major staple. Cassava roots and leaves contain two cyanogenic glycosides, linamarin, about 95%, and lothostralin, about 5%. When plant tissue is damaged, the enzyme linamarase hydrolyzes these compounds, releasing hydrogen cyanide. Cyanide inhibits cytochrome C oxidase, the final enzyme in the mitochondrial electron transport chain, preventing cells from using oxygen. Sweet varieties typically produce less than 50 milligrams hydrogen cyanide per kilogram of fresh root. Bitter varieties can exceed 400 milligrams per kilogram and, under drought stress, reach higher levels. Documented acute poisoning occurs when insufficiently processed roots or flour are eaten. Chronic low-level exposure is linked to neurological conditions. In sub-Saharan Africa, the disease Konzo, characterized by sudden, irreversible spastic paralysis of the legs, has been repeatedly associated with high cyanogen intake from bitter cassava during drought or food shortage. First described, clinically, by Belgian physician G. Trolle in the then Belgian Congo in 1938. Konzo outbreaks have been recorded in the Democratic Republic of Congo, Mozambique, Tanzania, and other countries. A 2017 outbreak in Kasizi District, Uganda, linked to cassava flour, produced 98 cases and 2 deaths, as reported by the U.S. Centers for Disease Control and Prevention. The plant's chemical defense evolved against herbivores. Humans had to invent ways around it. Indigenous Amazonian and later African communities developed multi-step processes that exploit the same chemistry that makes the root dangerous. Physical disruption, peeling, grating, or pounding, brings linamarin into contact with linamarase. Soaking in water for 3 to 4 days allows leaching of cyanogens and fermentation that further breaks them down. Hydrogen cyanide, being volatile, escapes as gas. Subsequent sun-drying, pressing, and roasting drive off remaining cyanide. Traditional West African products, such as gari, fermented, roasted granules, and fufu, pounded paste, rely on these steps. Laboratory measurements confirm substantial reductions. Prolonged soaking and fermentation can cut cyanogen levels by more than half, and combined methods routinely bring concentrations below safety thresholds when performed correctly. The knowledge was empirical, refined over generations by observing which procedures prevented illness and which did not. No single invention solved the problem. Accumulated practice did. The largest modern illustration of the danger is the repeated emergence of Konzo during periods of drought or conflict, when people shorten processing time or consume bitter varieties under stress. In the Democratic Republic of Congo's Kahemba district, A single dry season outbreak in 2009 affected nearly 2,000 children. Researchers, including those working with the Australian National University's Kanzo Prevention Unit, have shown that short soaking, 1 to 2 days instead of 3 to 4, leaves residual cyanogens high enough to cause the syndrome, especially when the diet lacks protein that aids cyanide detoxification via rhodonese. Portuguese introduction in the 16th century expanded the crop's reach, but the processing knowledge did not always travel intact or adapt quickly enough under crisis. If you're finding these documented links between environment, chemistry, and human health compelling, leave a comment with the most surprising fact so far, and consider sharing this with someone who thinks natural always means safe. Internet claims sometimes assert that all cassava is lethally poisonous, or that a central fiber is uniquely toxic. Verified data show a clear distinction. Sweet cultivars can be rendered safe by peeling and thorough cooking. Bitter cultivars require the full traditional sequence of grading, prolonged soaking or fermentation, and drying or roasting. The World Health Organization and FAO recommend a maximum of 10 milligrams hydrogen cyanide equivalents per kilogram in cassava flour. Codex standards set 50 milligrams per kilogram for fresh peeled sweet cassava root. Acute cyanide poisoning from properly processed commercial products is rare in regulated markets. Chronic neurological disease remains a documented risk primarily in regions where processing is incomplete and diets are monotonous. Claims that cassava is carcinogenic in ordinary use lack support from major toxicological assessments. The primary concern is acute and chronic cyanide toxicity. Uncertainty remains around exact dose-response thresholds for Kanzo in every population, but the association with high cyanogen intake is established by epidemiological and biochemical studies. Cassava began as a wild Amazonian plant defended by cyanogenic glycosides. Domestication, confirmed by genetic and archaeological evidence from the southern Amazon, turned it into a high-yielding tuber that could be stored in the ground and harvested as needed. Indigenous techniques of grading, soaking, and fermenting converted a chemical defense into a reliable food. When Portuguese ships carried the plant to Africa in the 16th century, it filled ecological niches left by less resilient crops and became central to diets across the continent. The same chemistry that once protected the plant against insects and mammals continued to demand respect. In times of plenty, multi-day soaking and fermentation kept cyanogen levels low. In times of drought or displacement, people sometimes ate roots after abbreviated processing. The result, recorded repeatedly since Trulli's 1938 observations, has been outbreaks of Kanzo, sudden paralysis that leaves survivors permanently disabled. One deeply human illustration appears in accounts from drought-stricken villages in Mozambique and the Democratic Republic of Congo. Families who knew the traditional methods still shortened them because hunger left no choice. And children, whose smaller body mass and higher relative intake made them more vulnerable, were disproportionately affected. Yet communities continued to plant and process cassava because alternatives failed more often. Modern toxicology has quantified the glycosides, mapped the enzymatic pathway, and set numerical safety limits. The plant remains a cornerstone of food security precisely because humans learned, through observation and cultural transmission, how to disarm it. The recurring element is the root itself, first a wild defense, then a domesticated calorie source, then a potential neurological threat under stress, and finally, a regulated commodity whose safety depends on the same processing knowledge refined over thousands of years. Cassava remains a primary staple for hundreds of millions of people, especially in Africa, Latin America, and parts of Asia. Nigeria is the world's largest producer. In commercial and regulated settings, processed products such as flour, starch, and ready-to-eat chips must meet maximum limits of 10 milligrams hydrogen cyanide per kilogram in many jurisdictions, including Codex and European Union standards. Fresh sweet cassava is limited to 50 milligrams per kilogram. Scientific understanding of linamarin hydrolysis, cyanide detoxification pathways, and the epidemiology of Kanzo continues to guide public health interventions that promote thorough soaking and dietary diversity. Where traditional knowledge is maintained and food systems are stable, cassava is safely consumed every day. Where processing is compromised by poverty, drought, or conflict, residual risk persists. Cassava was never a naturally safe food. It became edible only because human ingenuity matched its chemistry with precise time-consuming techniques. That knowledge is still the difference between nourishment and paralysis. What does it say about our relationship with the natural world that one of humanity's most important calorie sources requires deliberate detoxification before it can sustain us? If this story changed how you see the foods we take for granted, subscribe for more rigorously researched food history. Leave your answer in the comments, and share the video with someone who believes natural is always synonymous with safe. The truth is rarely that simple, and far more interesting